Hello, and thank you for joining IDEX Health Science today for our webinar on dynamic low-flow post-column mixing before mass spec analyses. The goal for today's discussion is to deliver some practical ideas for preserving flow path integrity and improving detection performance in LC-MS applications. We'll cover some recent challenges and describe a number of approaches to reduce carryover, minimize dispersion and band broadening that occur at very low flow rates, avoid sample degradation under extreme conditions such as very low temperatures, and of course always preserving samples. We'll also discuss how fluidic integration and control can be thought of as another tool to maximize system performance, starting with collaboration in the early development phases. You'll hear three voices in today's discussion. Two of us are from the Integrated Solutions Group at IDEX Health Science, and we have a guest we are tremendously excited to welcome. Our format for this program will be a brief overview and introduction of the participants, followed by a really galloping roundtable discussion. Toward the end of the hour, we'll answer any questions you key in while we're chatting. Start keying in your questions anytime into the box provided. Later in the program, we'll give you an email address to send us any remaining questions you may still have. I'll take a moment now for introductions. On your screen, you'll see some additional details about each person's fluidic experience and product specialties. Darren Lewis. Darren Lewis is a scientist by training, and Darren manages research and technology development for IDEX Health Science. Over the past 4 years that we've worked together, he has spearheaded quite a number of integrated systems we've released. Aside from combining science and technology, Darren's real talent is identifying exactly where technology needs a shove in a new direction to solve the fluidic challenges our customers bring to us. Very resourceful thinker. And it was Darren who recommended our guest, Professor David Schriemer of the University of Calgary in Alberta, Canada. They have a substantial development history, and in just a moment, I'll ask Darren to hit the high points on that. Finally, I'm Bob McCarthy, R&D Director at IDEX Health Science. Throughout my career, I've worked on a range of instrumentation, from cell culture systems to drug discovery systems in regulated environments. The reason to let me talk today is that a good portion of my career has been spent working in the field of mass spectrometry. Like all our webinars, this one will be available for playback on our website. You can find them all listed at webinar.idex-hs.com. One quick note before we get started with our roundtable discussion. We want these discussions to be open and informative for the engineers and scientists who are our customers. That said, because we deal with innovative and often patent-pending technologies, in most instances, we are bound by non-disclosure agreements. That means we won't be able to give you the names of recognizable partners or platforms, but the stories are real. Okay, Darren, remind us about the work you've done with Dave in the past and bring us up to where we are today.
I've been working with Dave for about the last 15 years. We started out in graduate school together at the University of Alberta. Dave's now a professor in the Faculty of Medicine at the University of Calgary. He studies proteins, structure and function, and basically focuses on the area of biological mass spectrometry. He also runs a core lab at the university. He's very practical, and he has come up with some very, very clever workarounds to biological problems. We started working together on a project years ago, identifying the binding constants between small molecules and proteins. Later on, Dave started working on issues of binding using H.D. exchange. I like working with Dave because he's pragmatic, he's very clever, he can always figure out a workaround to a problem. He's easy to partner with because he has a basic understanding of the science, and he also has a very good understanding of hardware. So you put those two together, and you actually come up with a pretty powerful partnership on development. Dave's also very good at describing problems to us when we are developing equipment. He can actually get to the root of the problem, and he's pretty direct when he needs to be. I remember a few times we've had some email exchanges when he didn't like the way some of the parts were performing. So we always get to the bottom of it. With his experience at the core lab, he also knows how end users use these mass spectrometers. He also knows how real samples need to get handled, not just ones that come from ideal standards. Today we're going to have a discussion about some of the limitations that present themselves when you're analyzing real samples using mass spectrometers and the fluidics on the front end, and some ways we can modify the fluidics on the front end to try and work around those limitations.
At IDEX Health Science, Our business day-to-day is the development of highly precise, very low flow, fluidic systems and components for scientific instruments. In order to do that, to stay out in front of where instrument design is headed, it's vital to work with scientists such as Dave to learn where the science is headed. That's what we're privileged to do and share with you today. So the online digestion application, what was involved in that?
We had a requirement of taking a gradient LC system, this is for protein separation, and processing the effluent in real time. And the processing essentially involved doing an online digestion step, and then feeding the results of that online digestion straight to the mass spectrometer. So in general, normal LC-MS type applications as applied to mass spectrometry, but the difference here was doing this digestion step in real time.
So Dave, just to remind me, the protein was whole protein separated on a column and then digestion, rather than peptide separated and worked on.
That's right, yeah. So a conventional approach to the analysis of proteins by chromatography would be to fraction collect. You would take the fractions out the back end of a column, prepare them as you need to, and then digest the samples individually, and then parse that onto the mass spec. Our requirement here, though, was to do this online in real time. And what we had discovered was that the digestion step could be enhanced dramatically by doing the actual digestion under mixed solvent conditions. So this would be with organic solvents, acetonitrile, methanol, isopropyl alcohol, and do these at elevated concentrations. Now, the problem is we need to do digestion for proteomics using trypsin. Trypsin performs really only at neutral or slightly basic pH. And what you've got coming out of the back end of a C8 column or a C4 column for chromatography is low pH solutions. So we've got an acidified flow, the organic solvent composition changes as a function of time, and none of that works well with a digestion concept.
So why couldn't I just do the usual thing and digest my proteins and then separate them? Why would you want to separate protein first and then do the digestion online?
It's an issue of complexity, Darren. We've got the reality in proteomics is our samples are really complicated, really complex from a compositional standpoint. And yes, although mass spec is a fantastic technique for arraying out multiple compounds, when you hit proteins, things are different. We really don't have the same sort of dynamic range, the same sort of ability to look at complex samples. And so really, in any sort of mass spec front end, you have at some stage a requirement for chromatography. So in order for us to achieve optimal performance on the mass spec side, we really needed to place up front an LC system. Now, we thought, well, why don't we fraction collect? You can, there's nothing stopping you from doing that, but you're introducing a lot of either robotic or semi-robotic type of fractionation steps. And there's losses. Anytime you handle a sample, particularly a small sample, there are the inevitable losses that you realize. When we discovered that digestion works really well under high organic solvent conditions, our first thought was, let's couple that into a flowing stream. But we had these practical realities to then deal with. And how do we take a solvent composition out the back end of a column that is dynamically changing and actually turn that into a constant or an isocratic feed into our little micro digestion elements? So these digestion reactors, as we call them, or micro reactors, they really function well when they're an extremely small bed volume. So when we approached you, Darren, our question was pretty simple. Help us design a fluidic system that allows us to take a variable composition and turn it into a constant composition by feeding in that extra stream.
So the scale of these feeds and the flow rates that we're talking about, can we talk about that just a little bit to give the context of the fluidics that we're talking about in terms of quantities, numbers?
Yeah, I think the way it worked when Dave approached me, he had a pretty reasonable range of flow rates that we knew we could hit. But what we didn't know was how well we could coordinate. The way the layout is on the equipment, actually, it had two separate gradients happening at the same time, one in one direction and another going the opposite way to neutralize the changes from the first gradient. So what we had to evaluate was somewhere between 0 and 5 microliters per minute, preferably 100 nanoliters to a microliter per minute flow rates, up to 1,500 or 2,000 pounds per square inch, I think, was about where it ran. How well these two pieces of equipment were going to interact as we performed a separation with one gradient and then counteracted the effects with the chemistry of that on the protein digestion. So what we had was, in the end, I think, Dave, very, very good precision that allowed it to happen probably within 0.5% precision run-to-run.
Right. When we first tried what we've been calling this mirror gradient concept, and so the application of this second gradient on the back end of the LC column was to essentially reverse the gradient from the LC side. What we tried at first was to just simply swamp that with a high flow of a second solvent. And, of course, that really isn't going to be an effective way of doing that. So when we approached you with this mirror gradient concept, we really needed to achieve that level of stability. We tried other pumps that were available to us at the time and tried to couple them together ourselves. And we could see that the concept was sensible, but we could clearly see that the solutions available to us were not going to cut it. We needed help.
I'm not going to cut it from the standpoint of stability, or?
Well, we couldn't use the system, right? So we could mock up 10 trials, one that worked, but it was beggarly difficult to actually get 10 out of 10 tries, essentially.
So I remember when we started working on this project, we had a pretty good specification from you. We had the experience with the hardware, and we were able to kind of automate the interaction of these with firmware to do the control. We learned a lot from it that we managed to push in the other hardware, but we did a lot of back and forth with you on that. In the end, I think we got down to a tolerance of something like 0.1% reproducibility.
That's right, and I think that was essential. Anything other than that, what we found was that the lifetime of the digestion column really suffered. Every time we took a hit from a mixing standpoint or from a stability standpoint, it essentially reduced the amount of active trypsin in the flow path. So what we found in the end is when we had it optimized, we could run this system without stop for dozens and dozens of runs with no obvious drop in performance. So we got it right. And the essence of that was achieving this really micro-scale or nanoscale titration event that we needed. I think anyone could pick up components to do this, that, or the next thing. But when you have to get everyone singing together off the same song sheet, that's more difficult. And most academic labs, or even I would imagine corporate labs as well, they're not going to invest the time and the effort into software development until they know what the product is or what the concept is. When you've got this feeling around phase like we definitely had, and at the same time a requirement for software development, that's where you guys really helped us a lot. You could kind of react with us and develop the things on the fly that we happen to need.
Yeah, and I think that the real advantage that you got with this over syringe pump, and I think it's fair to say you wouldn't be able to do this with a regular syringe pump.
Well, no, and so one of the hidden secrets with syringe pumps is that they're fantastic at delivering fixed volumes as long as you use the entire stroke of the syringe. But they're absolutely horrible in flow rate precision. For these sorts of applications, you get peak-to-peak variation of around 20% or so, and you just can't even imagine doing an experiment like this with technology like that.
And I know there's some other artifacts that come up, right? If you have anything compressible in the flow path and you have to build pressure, you have variations in achieving the set point. You have air bubbles in there, you have some other trouble. We've actually managed with our flow feedback system with flow sensors on our pumps to manage to compensate for all of those in real time, which I think really does give you the precision that you're looking for.
Okay, so you're saying that syringe pumps really do have a place, but maybe not in pressurized.
Yeah, I think it's fair to say there are a lot of very good applications for syringe pumps, just not necessarily nanogradient work and not necessarily work where you have an outlet that varies in pressure or where air bubbles could become a problem for you.
And so that was an issue for us too, right? The outgassing of the solvent when you drop things across a pressure drop like we did or flow things through a pressure drop. That's what required us to also operate the digestion element at an elevated pressure as well, so we could control that. And that was part of getting that extra level of performance.
We work with a lot of people who do mass spectrometry, and I remember thinking about some of the early problems that you were probably going to encounter related to actually doing the ionization of this sample downstream, applying voltages, and concerns about things like dead volume.
Right.
And I think that probably helped to color how we laid out the instrument and how we operated it when we worked with you. I remember going on site. I remember we did quite a few experiments on site in your lab, and then I did some back in my lab to kind of validate where you collected the sort of real-world data, and I did some backup validation of it. And I think that partnership is a pretty ideal type of relationship to have between an equipment manufacturer and a researcher.
The Controlled Delivery Module, or CDM for short, is a compact pumping subsystem that provides precise flow control. Through feedback from an integrated flow sensor, the CDM regulates flow rate by using an algorithm, dynamically adjusting pump speed in near instantaneous response to changing flow conditions. The CDM incorporates a pump, high-pressure rotary shear valve, flow sensor, and controller. It comes fully tested, ready to plug into your application and evaluate right out of the box.
Do you think that two gradients operating in opposite directions with ultra-tight control and precision and elevated pressure is probably about the most difficult thing you could have picked to do?
Well, probably. It certainly wasn't something that was thought of, largely because when we think of LC-MS systems, our idea is to do the least amount of fussing about, if you will, from the back end of the column to the front of the mass spectrometer. And we learned that through hard experience, right? We have sample losses if the distance is long. We reduce peak concentrations if the distance is long. We make a horrible mess of a peak tail. All these things have led to the lore in the field that you really shouldn't be touching anything post-LC. And I would rather say you really shouldn't be breaking any of the accepted rules in fluidics post-LC. And so our system with you, that we developed with you, allowed us to achieve, in fact, better peak definition going into the mass spectrometer than we would have just by coupling the LC directly to the mass spec.
Yeah, and we might want to explore that topic just for a second, too. Common convention, when you take LC in school, they talk about not doing anything to the fluid downstream of the column. So a perfect system would have column separation going straight to the detector or the electrospray, whatever you're going to use. But what you've done is you've added all kinds of things in there, and you're saying you still didn't get peak degradation, or you got a minimal amount?
A minimal amount. I mean, no one ever wants to claim by putting things on a flow path that you're not doing something to the system. But what you really want to avoid is asymmetric peak broadening and excessive peak broadening. And if you can achieve that and yet at the same time improve your detection efficiency for the analytes of interest, you've actually won, and that's what we've achieved. So online digestion allowed us to turn proteins into peptides, and that allowed us to achieve, first of all, higher sensitivity, but also it actually improved, in this case, the peak definition because we could do it in such a way that we avoided a lot of the absorptive loss that you experience with looking at intact proteins. So we have a couple of figures in some of the papers that we've developed where it actually shows that the peak width of the peptides that we get from this total processor that we developed are actually narrow than if we turned off, quote unquote, the digestion part of it.
That's a really interesting consequence, and I think we've seen this on a few other projects we've done where we've worked with folks on introducing reagents ahead of the mass spec, is that conventional thought is that it always will make things worse. In a lot of cases, when you take care of things, you do it properly, you enhance the performance of the machine overall. You do a much better job of what you're trying to do.
You definitely do. And so I think one of the things that helped us in our particular situation is we could, at one point, reacidify the flow before it actually went into the mass spectrometer. Mm-hmm. And so we actually had 2 points of addition post-LC. And you need to be willing to think outside the box that way, right? You need to be willing to ask, what can I do to improve the detectability without restraining how you improve the detectability?
That's exciting for us because we make all kinds of little gadgets that can go on the front of a mass spectrometer or in a fluid path. So the more exploration you do, I guess we're probably a lot happier.
Right, absolutely. Well, I mean, everyone has a parts drawer that's filled with upchurch fittings. And, you know, one of the things that we, of course, had to suffer through is trainees and technicians using the wrong parts for the wrong thing. And we all do that. We like to cobble together things. And when it doesn't work, we kind of blame the concept. And then a lot of times we should be blaming the implementation of the concept, or not necessarily who did it, but what combination of parts they used. So working with you folks was actually very useful that way. We had these discussions about fluid path design rather than just say which part in the catalog do you want. So that dynamic was good.
You know, I wish I could tell you that I've never used the wrong part in the wrong place before as well. There's a lot of experience you gain by doing it. Well,
and so I don't also want to suggest that I haven't screwed up either. There's a term that we use in the lab, and it's called Schreimerized. And that's when you take the fitting and then you over-tighten it.
So the good news about that is ordering new parts is very easy. So you can go ahead and do that.
Oh, okay, so Dave, why does proteomics need this? I mean, where does this fit in the grand scheme of things?
What we wanted to achieve is the development of a proteomics workflow that allowed us to do what we call both bottom-up proteomics and top-down proteomics. Bottom-up is the classical approach of taking a mixture of peptides and letting your LC-MS do all the work. Top-down is taking the proteins and going directly into the mass spectrometer. If you want to switch from one to the other, that is a complete pathway change. You have to handle the samples differently. You have to approach all the experiments differently. We wanted one fluidic system that allowed you to have software control over that decision. You want to look at proteins? You look at proteins. You want to look at peptides? You look at peptides. So you inject your sample into the system, and you don't care if you're doing top-down or bottom-up proteomics. You can decide through software. In order to build a fluidics pathway that way, we needed to encompass an autosampler for sample introduction and all the valves associated with that, an LC system for protein separation, obviously the mass spectrometer, but what the key was for this entire concept was the insertion of a software switchable digestion technology, and that is between the LC system and the mass spectrometer itself.
And let me guess, they're not off the shelf.
Nothing is really off the shelf in our lab, fortunately or unfortunately. So we had then problems. We had obviously LC techniques that we could pull off the shelf in that case. Those were the easy parts. But we had to deal with all the issues of pushing protein through a very complicated flow path.
Yeah, and I think that when we took on the project, when you started working with us, the biggest problems that you had involving chemistry, you had already sort of taken care of a lot of those. And what you were having was a real practical issue of control, getting precision and accuracy so that you could optimize the process, right?
That's correct. We knew what we wanted to achieve. We just had no in-house ability to do that. And that's the frustration that we experience a lot of times in technology development, in research. The concepts are there, but testing them and refining them requires a level of commitment to engineering and software development that in many cases you don't have yourself within your own lab.
Yeah, it made a great partnership because I remember we focused a lot on the fluids, on the precision, a lot on the control, optimization, and we could rely on you for all the chemistry and the background, the biology. In the end, we came up with, I think, it was a pretty nice piece of equipment.
Well, sure, but don't overlook the fact that you understood the language we were talking about, right? We could explain the fundamentals to you and the issues that we thought were relevant or germane to our overall objectives, and you knew where we were going. And that was, I think, an important part of it. You knew why we had to do certain things a certain way. And so for us, that made the process a little easier to get through.
And if you had done MALDI, Bob would have been far more interested, of course, because he's got the MALDI background.
Yeah, the movement of the fluids in that is a lot less exacting, I would say. You can dry a spot onto a plate and go from there. But the fluids here, so I really love this thought process of the stability and the flow. So are there other techniques besides the ones we talked about, pumps or other things that you have found as would have been adequate for this? I mean, I understand about the controls, but specifically the hardware. Anything you have seen that's different about ours?
Well, what we found was when we first started the development of this concept that we really, really had tight tolerances. We did not have a lot of room for error. So when we considered all the various other options that were out there, really all you had were twofold. You have an LC system where you swamp the flow out of the LC, in the back end of the LC, so that if you've got an acidic solution coming through, if you crank up the volume high enough, the volume flow rate high enough, you can remove all your problems. But, of course, then you definitely dilute your signal. And so that was anathema. We were not allowed to do that sort of thing. We try then to apply this gradient concept that we talked about before, and we found that unless you had tolerances or precisions on the order of 0.1%, you just were not able to achieve the performance that we needed. We got wild pH swings outside of that tolerance range.
And I think, when you look at conventional pumps, actually look at any given pump. I've done a lot of work with pumps, and pumps have several different causes of flow rate fluctuation. So the main one that you see, anytime you have a drive mechanism like a lead screw, you're going to see variation in the output flow as the mechanism turns. That's a natural consequence of manufacturing processes. Any pump that doesn't have variation, it probably will jam. So that's sort of the tolerance between a thread and a nut, essentially, as things turn around. That one's sort of the dominant, lowest frequency flow rate fluctuation you get. You add on that higher frequency fluctuation that come from things like stepper motors turning, finite resolution of those steps, damping effects of the steps, and then also environmental effects like temperature changes. You start to wind up with a situation where, particularly when you get below a microliter per minute, you find it very, very difficult to get absolute stable control.
And that's what your flow sensor devices helped us appreciate. Remember, a mass spectrometer is a concentration-sensitive detector, so a lot of times you're not going to see the impact of a flow rate change unless you start to do creative things in the flow path like we're doing. And so even just a simple action of putting your hand over, not touching, but putting your hand over the flowing silica tubes connecting to the mass spectrometer, you could see that change the flow rate.
From the temperature.
From the temperature, yeah. So that, of course, launched an entire discussion about how we have to make sure that we know exactly where our fluid lines are going, and we control that. These are things that the practitioner in a lab often doesn't do when they're thinking mass spectrometry. It's uncontrolled spaghetti.
So you want a set point stability that environmental disturbances or, like you said, manufacturing tolerances changes do not affect. You want that worry taken off your list of concerns, right?
Right, and that's what we managed to achieve in this relationship. We understood where all the concerns were and how to address them properly. And it's, I think, in any good area of science meeting engineering, you need to know what is an issue and what isn't, what to design around, what to spend your time thinking about and working towards, and what not to. You can spend endless days and months, as you well know, fussing about the wrong problem. Mm-hmm. In our case, we could get to those problems pretty quickly.
So for the online digestion work specifically, the summary of where we are, we're talking about low flow rates, and we're talking microliter per minute range and into nanoliter per minute range, right?
Yeah, probably, I think, 100 nanoliters a minute. In a practical sense, 100 nanoliters a minute up to 500 or 600 nanoliters a minute was probably the most common.
That was the most common. I'd say 500, 600 nanoliters a minute. We certainly ran things up higher.
But remember, it was a gradient, so the total flow rate would be 500 nanoliters a minute, which means the B pump would be on the order of 10 or 15 nanoliters per minute when you start going.
That's right.
It would increase its rate up to maybe 80% of the total flow.
Right. So we had the LC flow, say 200 nanoliters a minute. We had the mirror gradient flow, another 200 nanoliters a minute, and then we had the reacidification flow at around 100 nanoliters a minute.
So do we talk about percentages, precision at that low flow? That's a pretty tall order, getting down to, 10 nanoliters per minute range and have it be really stable. All those effects are noise on your system.
I would say the reproducibility is extremely, extremely high. Yeah. That's how I would describe that, because you could be at 15 plus or minus 1, right, but it's exactly plus or minus 1 every day or whatever, and that variation averaged it out over the course of half an hour or 20 minutes. So it kind of worked out. If you were ever trying to commit that I can give you 15 nanoliters a minute, plus or minus 0.1%, you're on crack. You're only averaging that.
That's right.
At any moment in time,
that's
it
exactly
is that, right? So that's sort of the issue with that.
Mm-hmm. So then, summarizing this online digestion work, the main requirements, Dave, what would they be in your mind?
I think it would be achieving a system that can do more with less. Our whole driver for this concept was to recognize that there are many situations when you only have one single sample, and you cannot do X, Y, Z to it as you would, say, in a classical workflow where you have the ability to do one type of analysis on dozens or hundreds or thousands of samples. We've got one quivering little sample. We've got a graduate student who spent months spending, lab time preparing the sample, and you want to get the most information out of it. So for us, the whole idea was to achieve the ability to do top-down, bottom-up type analyses on one sample with a minimum amount of manual sample handling.
I think that the main thing that's been sort of discovered through all this is that you can do a remarkable amount of work on a sample, even after you've separated on a column, and still not destroy the integrity of the sample. I think that's sort of the main main thing here.
Not necessarily intuitive to people that have been working in the field for a very long time. And those are areas that used to be no-nos in a lot of ways.
That's right. Right. We're in a little different world now, I think.
Mm-hmm. Over the years, we've streamlined the process of collaborating with our customers to shorten the development time between initial concept and manufactured solution. The most successful partnerships begin with instrument designers willing to share their instrument schematic to let us visualize their whole fluid path, not just the nagging problem in the corner. Most are surprised at the level of support and discovery throughout the development process. All right, so looking to the future, let's talk about a couple new applications.
We certainly have other projects that demand fluidic solutions or fluidic components. And one that our lab has been interested in for some time now is in the area of protein structural analysis. Now, I know we've talked about proteins and proteomics already, but that's the science of looking at what proteins are in your sample and what's their sequence. This is a different application. This is more three-dimensional in the sense of how is a protein constructed from a shape concept, what is the topology or the topography of a protein, and how does it change, either as a result of a drug binding to it or another protein binding to it. It's really a better connection to the function of a protein than the proteomics that we talked about. So this is an area that has been around for some time, but in broad strokes, it's essentially this. You use heavy water, or D2O, to paint a protein. You paint the protein is simply done by adding D2O to a solution of your protein, and that D2O exchanges with amide hydrogens. And so obviously a protein is constructed of amino acids, and when an amino acid is bonded to another amino acid, you have an amide bond. And that amide bond presents a hydrogen to the outside world. Now, when you add D2O to this sample, you have the opportunity of exchanging that hydrogen with a deuterium. That's a dynamic event. And really, it's probably easiest to say it is a way of understanding what part of the protein is exposed to the solvent and what part of the protein is buried within the solvent, and how that changes as a function of time. So really, it's, as I mentioned, it's a way of painting the surface of a protein, and once painted, you can stop the painting activity and then process that sample and find out where your paint went and what changed in the overall picture if you happen to use another drug or a protein in that sample.
So to be sure, to understand function now, you're talking about a typical application where that would be utilized.
Sure. So HD exchange is a technique that is certainly used in the research community for understanding what we define as the protein folding problem. How does a protein fold or adopt its overall three-dimensional structure? That's of interest to some of us. It is also of interest to understand what happens during the life of a protein. It interacts with other things. What happens to the protein when it interacts? Does its shape change? Where does the interaction occur? It's really that connection between structure and function. And mass spectrometry really wasn't the tool classically used for that type of foray. Normally, you're going to use X-ray crystallography, NMR, all the conventional tools that you can imagine using for an exploration of protein structure and function. HD exchange and related technologies have really taken an uptick lately in terms of its utility for looking at these sorts of questions. The real niche area is in looking at large systems, looking at large complex protein systems, and And this is not just the research community that's interested in this. This is the pharmaceutical community using it as a technique to look at antibodies, how antibodies bind to their antigens, and discovering what the actual epitope is, mapping out the context, and really, from what I understand, understanding when you have an antibody that's different from your competitors. So it's useful in the whole biosimilars environment. There's been a lot of interest. We've certainly had a lot of interest, not just from other academics, but also from biopharmaceutical companies who are exploring the use of mass spectrometry in this fashion.
In fact, I think there's commercial equipment available for doing this, right?
Yes, there are. A number of LC and mass spec vendors are quite interested in fielding technologies for this, and I believe a couple of them do already.
So do you think there's an opportunity, is there a hole missing in the product offering now? Could we extend the type of work we did in a previous collaboration, create a piece of equipment to meet some unmet need?
I think so. One of the things we need to recognize is that even though we talked in our previous example about proteomics and a fairly complex flow path, we can't let ourselves think that that's the end, that we should stop our flow path programming and, oh, that's too complex, we need to back off from it. There's a lot that we can do armed with the knowledge that we've gained and that you folks have gained. In HD, we have other complexities. We need to be able to analyze smaller samples. Currently, we're wasting a lot of protein, using far too much in the way of sample. We need to also come up with a system that is quicker, I would say, than our current product offerings in the field and what we can achieve ourselves in the laboratory. And we need to recognize that there's extra complexity added by the temperature requirements. We need to do our processing at 0 to 4 degrees, essentially, and that's not an area where you like to do chromatography. But again, staying in theme here, why not? If your application needs it, let's find an appropriate solution to that problem.
Well, it's a great fit when it comes to micro nano, so I think we've got that one well covered. I think what we would probably have to do to move forward would be to really look at aspects of the flow path and how you'd like to optimize it. We've got a lot of familiarity in working with you on that from our previous relationships, so I think we'll be able to figure out how to work together on that pretty easily.
So the things we've learned from parsing fluids into and out of enzyme microreactors are going to be really useful. We know how to do that well, and we have probably multiple enzymes that we would want to consider using. And I'm not saying that all this has to be embedded in a lab-in-a-chip concept. I think, again, that's one of the things we've learned is you don't need to do that. And when you do that sort of thing, obviously your throwaway part becomes that much more expensive, right? So I think we're both sensitive to the fact that build the right pieces into the flow path and avoid the needless complexity.
Yeah, I think there are some common pitfalls that people see when they build equipment, things like dead volume that you get when you start to use a part in a way that forces it to be used differently than it was intended. So a chip, for example, trying to do electrospray from a chip, very challenging sometimes. You start to add a lot of parts to get that done, and suddenly you have extra volume.
Right.
So sometimes you can actually get the same thing done using a much more simple method. Sometimes you need to have the chip in there, but you have to make sure that when you incorporate it, you've got everything figured out in terms of how the fluid's coming in and going out, or you've really undone all the good work that you've done by having a low internal volume component.
That's right. That's definitely right.
So I've looked into this a little bit. It looks like the addition of the D2O, followed by cooling, and then some chromatography, right?
That's essentially it. The devil's in the details, though. The current configuration that you can buy or that you would imagine doing yourself in the lab is largely autosampler driven. We have a situation where the D2O is added to the protein, it's incubated for a certain amount of time, and then it is quenched also by an autosampler with a low temperature acidified solution. And then that sample, again, driven by an autosampler, gets picked up and placed into an LC-MS system.
So those are all commercially available. You can do that in your lab just with the equipment you have.
That's right, and that's what we do. And it's an adequate solution, except if you want to achieve better performance on smaller amounts of sample. We are limited in certain situations. And for us, the limitation comes in terms of the amount of sample that we're required to use. It's frustrating when you use an autosampler. There's always waste. There's always carryover. These are still driven by syringe pumps. We know the problems with syringe pumps. So those things carry over from our previous experience. Our biggest issue right now is clogging syringes when we're trying to use small volumes, and these small volumes are really only as low as 10 microliters. This isn't really a small volume as we define it in the field. So what we want to be able to achieve, Darren, is get our protein sample into a fluidic path and do everything there. Do the labeling with D2O, do the quenching and acidification at reduced temperatures that's required, and then all the LC-MS work. So our bigger challenge here is one of synchronization. We have some ideas on how to achieve all that in a fluid path, but we'll have a larger problem with synchronization than we did before, so we'll need to talk about these things.
Do you think the miniaturization is going to be as big a problem as synchronization? It sounds like, in terms of scale, it's similar to what we've done before.
I think so. And as we approach the problem before, we'll start at a certain scale, understand the fundamentals and the issues that present themselves there, and then leverage our knowledge on the nanofluidic side to drop things down in size. And we're so beholden to you folks at that level. There's only so much we can cobble together. We've done as much as we can in that regard, but now we'll need that combination of systems control and miniaturization that we just simply can't do in our own lab.
One of the things that we've been working on a lot are embedded controls, trying to figure out how you want to synchronize these in an efficient manner, get all the timing right, have everything talk correctly.
Right.
We've got a good handle on that. We'd obviously have to look at, for your application, whether what we have right now is a good fit or we have to do more optimization. But if it's anything like the projects we've done before, I think we have a pretty good fit to start a collaborative arrangement, collaborative relationship between us, where we can take some of your ideas, try out a system, maybe get it in your lab and work with you on that.
Right. I wanted to bring up that whole concept of embedded controls and gluing the system together, as you said, looking for something to address these challenges to get things online. Just to juxtapose it, if it weren't for having a system delivered to you that was already tested in your application, or at least as near real world as we could get, what would you have to do? What would be the way you would do it in your own lab?
I wouldn't attempt it. I think there are many labs that have wasted time and resources by being overly optimistic in terms of what they can achieve. We need to appreciate the complexity that's involved in this sort of undertaking and this sort of foray. If we don't have in-house capability, people who understand the fundamentals, people who know fluidics, and people who know what is achievable on the engineering side, why would you start a project like that? And why would you not then approach your science differently? And there's always that option to do as well. But we've embraced the fact that in order to address some of the current issues or the frontiers in this particular field, we'll need an engineering solution before we can go much farther. Yes, we could try to do it ourselves, but I think we'll fail miserably because the problem is ultimately an engineering one, and we know that.
We obviously really love this opportunity to work early on in these discussions in the applications. I think for me, the embedded controls story isn't parts. It's no more just parts than if you were to buy just a pump from us. The idea that we love about this is working with you at this stage of a development project. And rather than have a spec that you send us, find out what you're trying to accomplish and really have a lot of give and take back and forth between the groups. And that's not just Darren. As it turns out, to your point, we're hopeful that, sometimes you're having folks in your group talk to our controls folks and, and people that wear a lot of different hats, but have some specialty instrument development capabilities and software, firmware, and even some, in your case, chemistry applications experience.
It can be a frustrating relationship at times. I've had experiences in the past where you try this sort of thing, of integrating your concepts and fundamentals into something that requires an engineering solution, and it doesn't work. And you sit back and you ask, well, why doesn't it work? And it can be for a number of reasons. A company might not be engaged in the problem. That's not you. You are a company that's interested in the underlying fundamentals, and you have a futuristic look. That's one of the things I know that Darren brings to the table in particular. And those are essential ingredients, and you need to be able to know that you can pick up the phone and be only like 1 degree of separation, maybe 2 degrees of separation from someone who's got the ability to comment cogently on what it is you're attempting to do here. And so that's just human relations, and some companies get it better than others, and it's been a real pleasure to work with IDEX on that.
I think also just one more thing that we don't want to underestimate is experience. So we've figured every way not to do fluidics correctly. And I think now we understand if you want to take a sample, do some processing with it, and eventually wind up doing electrospray, we've actually figured out a lot of the pitfalls there. We work with a lot of customers on problems like that. So we can, I think, a lot more quickly come up with solutions for you that are more likely to work and suffer a lot less of that deterioration that we see. So I think that's really a big advantage of doing this type of collaboration. We can provide that kind of insight, and you can focus on the science.
And I like that the telephone numbers go both ways. I mean, really, it is a collaboration. Now, we're learning every step of the way with you. And the fact that you're not expecting us to be experts without ever a misstep here and there that we can correct quickly together is really the heart of it for me.
Right. There's an appropriate attention to the legalities of it that I found, in terms of what's confidential, what isn't. There's an appreciation for what's sensitive and what isn't without burdening the whole process. And I think we can all point to those sorts of examples in our past where good ideas were overly burdened at the front end.
Inside our customized modular designs, you'd expect to find industry-leading components. What you might not expect is the extra attention to integration, control, and validation testing that saves you time, money, and effort. Nearly every one of our integrated fluidic systems ships with a unified control interface that makes our systems easy to use, control, and monitor. One more thing you don't need to think about. So if you haven't had the time yet to request the idea book, take a moment now to jot down this URL to download your copy. Darren spent more than a year working with our technical teams, product managers, and customers on the applications and system configurations in this book. It's completely jam-packed, so please don't miss it. We obviously enjoyed discussing this topic and certainly could go on, but it's time now to welcome our online listeners into the discussion and answer your questions. So thanks to those of you who have sent in a query. I want to mention that a couple of the questions we see here are related to specifications that we may not know off the top of our heads the answers to. So please be assured that you will be contacted soon with answers if not in our live segment here. So on to the questions. Dave, you're our guest, so I think we're going to dish the first one to you, if that's all right with you. And Darren and myself will always argue and contradict if we see fit, as usual. So the first one goes like this, Dave. How does the digestion box work? Like a column or what? It's a question that I think was referred to in our diagram about the top-down and bottom-up.
Right. So I think this is a question specifically addressing the triptych digestion workflow for proteomics. And it's really we call it an enzyme microreactor. So a small cartridge, essentially, that we like to operate really as a column. So it has all the features of an analytical column. In this case, though, your stationary phase is protein.
Okay, next one. Darren, I think this one I can dish to you. This one goes like this. Why are things more different at nanoliter flow rates, and why can't I just miniaturize things that work at mil per minute flow rate?
Well, it's a good question.
Yeah.
Truth is, you can just miniaturize. The trouble is all the things which aren't a nuisance at higher flow rates, like trapped air bubbles, surface area of tubes where things can absorb, mixing volumes that get swept through very, very thoroughly, suddenly become much larger impactors on the flow performance when things are smaller. For example, a 1 microliter air bubble takes time to compress. If you're flowing at a milliliter a minute, it takes just a few seconds to compress that air bubble and have your flow rate achieved. But if you're flowing at 100 nanoliters per minute, a microliter air bubble has a significant amount of time to actually squish down before your flow rate gets met. And it's the same issue with effects of temperature and a variety of the other environmental things that affect flow. So you have to look after all those in greater detail if you want the same relative amount of precision of flow that you expect from a high flow system. So in general, you can shrink down, but you really have to be careful about things like surface area and bubbles and compressibility and a number of those other things.
And I would just add also that, our experience has taught us that when you get down to the lowest flow rates, we can typically really monitor microliter and below. Really, we're talking about precision, and you talked about it earlier in the program today. You can't talk about accuracy at that kind of flow rate because you're talking about the limits of the measurement devices and a number of other things, even in terms of your measurement precision. So I think what we're saying is that at this flow rate, the flow sensing devices that we couple to our pumps do really well at doing that kind of, precision and stability that Dave talked about in the online digestion work.
Yeah, absolutely. I think, just to follow that up a bit further, you raise a valid point. It's very difficult to get a gold standard at 100 nanoliters per minute largely because of all those environmental effects, whereas standards for flow are much easier to achieve at milliliter per minute and above flow rates. So you're right. For most applications, it seems like precision is the name of the game.
Yeah. So let's move on to another one. There's a question here for you, Dave. When you're doing HD exchange, do you always have to have a model for it?
Well, it's not a requirement for the method itself. You can certainly do HD exchange on a protein if you don't have a structural representation or a model for it, as the question suggests. However, when you do have that model, you can certainly understand the results a whole lot better. You know structurally which regions of the protein are either close to each other or remote from each other. So then when you're actually measuring the D2O or the deuterium uptake in a specific segment of sequence, you can then localize that on the structure. So the model does help, but there's a small number of us who are actually considering ways to take this data and actually use it to model structures, so a true de novo type structure determination. I think that's really one of the exciting future applications of the technology.
All right, next question. How does the complexity of integrating a syringe pump compare with a piston pump? This is a good question. I'll start the fielding of this, and either of you can jump in with me. We talked quite a bit in the earlier program about the syringe pumps being really good at a number of things, and Dave basically talked about the fact that if you use the full stroke, they're quite good at what they do. But if you're doing the kind of gradient work that we've discussed, and you want to use part of the volume of the pump, for instance, and you want to control that flow very precisely, there are some advantages to a piston pump with especially some feedback control, which we've talked about. So we have been able to do much more effective mid to modest pressure range flow feedback with piston pumps that are the Sapphire engineering pumps that we developed, and couple that into systems in some ways a whole lot easier than using the full stroke of the syringe without that flow feedback. So we feel that there's advantages to that. The pumps themselves have a stationary seal and have quite long life, so there's a little less maintenance involved since there's an awful lot of spaghetti typically in these LC-MS systems. Reducing that complexity sometimes helps as well. Any other comments from you, Darren, on this one, on the syringe versus piston?
Yeah, I think if the question's about, relating also to the complexity of the physical integration parts, like the communication bus and protocol and how easy the command structures are to set up and things like that, I think that all depends on the driver that's used to run the pump. So typical, commercial syringe pumps have a driver that comes with them, a board that's in it, and some command structure that you're given. It talks typically on a serial port. Well, on our pumps, if we provide the driver, of course, they're pretty straightforward. We actually have fairly complex command structure that has all different levels depending on how much control you really want. We offer quite a bit of flexibility with that. But if you just order a pump without any driver card, of course, it's up to you to do all that kind of integration. So I'd say in general, the rule of thumb is that there isn't really a difference. There doesn't need to be a difference in complexity between the integration of a piston pump or a syringe pump, depending on what you want to do. And that's more on the control side of it, right?
Yeah, sure. Yep. Yeah, it's certainly a combination in all cases in these applications of trade-offs for hardware and controls, I would say. Mm-hmm. All right, so next one. Here's one about pressure itself. So we hear much about going to elevated pressure in LC in general. Would this enhance either of the applications you outlined in this program? Dave, why don't you want to take a first crack at that?
Yeah, certainly. I think we have to understand why people are going to high-pressure LC systems. And that's largely to take advantage of smaller chromatographic particle sizes so that they can achieve more efficient separations, higher resolution chromatographic separations. And as a requirement of these smaller particles, you need to apply your flow at a higher pressure so that you can get the flow rates that are optimum for that kind of design. So for the applications that I'm looking at, the issue of pressure is different. Let's consider the triptych digestion first of all. There I can definitely see where higher pressure is good, but not for the reasons I started off with. In this case, it would be for helping in the denaturation of the protein, which would lead to a more efficient digestion of the protein. So there are researchers who do apply large pressure drops across these enzyme microreactors on the order of 10,000 or 15,000 psi, and that actually markedly enhances the digestion performance. So I think that's an area where higher pressure could be very useful. In the HD applications, that's where you want to be careful with pressure. Remember, it's a technique that helps us understand the three-dimensional features of a protein. So if we're applying high pressure, we need to worry about how that high pressure is changing the three-dimensional structure of the protein. So we can use it after the quench happens and after the digestion step as well, I would say. And in that particular scenario, you could then use it simply to get it to the mass spectrometer faster while achieving a base level of performance in separation. So different issues. There is a role, but it's somewhat of a different combination of effects than is typically involved in high-pressure LC.
All right. Well, another one. We get probably time for just about one more. So this one is sort of related, I think. Can you comment more on the difficulties encountered with mixing at really low flow rates? Darren, you want to start that one off for us?
Yeah, I think that I guess my take on that question would be, what are some of the deleterious problems you get with accidental mixing at low flow rates? For instance, if you're rotating an injection valve and you sweep past a port that has some material in it, how much do you need to worry about that? Well, generally, like I talked about in an earlier question, at lower flow rates, those become more troublesome problems. At high flow rates and large internal volumes, you can tend to sweep things out better, and a little tiny bit of material that carries over and mixes in becomes a much less significant effect. But when you shrink down and you're flowing at like 100 nanoliters a minute, having 100 nanoliters of material in a port somewhere that can actually sweep in and mix with your sample becomes much more complicated problem. And you have to be much more careful about trying to ensure that your pressures are balanced when you switch valves or when you switch from column to column or you do some other kind of problem like that. Also, the post-column effects that we talked about in the webinar earlier on are a lot more significant as well. And maybe Dave could speak to some of that as well.
Yeah, I guess my concern would be more in achieving effective mixing of reagents that are essential for the processing that you do along the entire fluidic pathway. For example, when we talked about the digestion step, when we have to do a pH adjustment, that mixing has to be very effective and rapid within the time scale of the analysis. So, when we go to lower flow systems and we drop into laminar flow rate regimes, that mixing is diffusion driven, and it takes time for diffusion to happen. So what we need to achieve there then is probably a slightly different flow path in order to get that to occur. Or what we always like to say is sacrifice the first little bit of your column bed to achieve that mixing. Columns are very good mixing elements, so those can be inserted in your flow path judiciously.
Well, we're right at the time block here that we have to wrap up, and we'd obviously like to go on for a long time. Again, we will answer questions that weren't answered live here very soon online, and please forward any other ones that you have for us. To review all of the archive segments in this webinar series, visit webinar.idex-hs.com. Once again, take the opportunity to download the idea book. This one is one you'll use over and over. For additional questions, you can contact our guest, Professor David Schriemer, at dschreim@ucalgary.ca. You see that spelled out there. And then, customer service for those of us at Health and Science, customerservice.hs@idexcorp.com. And finally, our great thanks to Dave for being our guest and a pretty good sport about it. For IDEX Health and Science, I'm Bob McCarthy. Thank you very much for joining.